First, let me thanking the ILSI Europe for inviting me to join this fantastic meeting. And when I was flying from Europe to US, I was remembering the story of Christopher Columbus. Because he started also from Europe and he was going to discover a new area. And in fact, he was taking advantage of a new technology, which was the caravel. Maybe you remember the name of the caravel, maybe not. And in fact, he was also looking for a new way to look at something. And in fact, if we look at the field of gut microbiota, we are realizing that food is there to nourish the host. You can see the gut microbiota on the diet. First, looking at what we call disappearance data. For example, people were giving the same amount of energy to two groups of people with different microbiota, and they were looking at the energy output. Or they were looking at the energy intake and the output of metabolites, and where the metabolites similar depending on different gut microbiota. Or they were looking at the global consequence, energy in, and where there are some people becoming fatter or leaner. All of that has been presented in a workshop that was chaired by the fantastic lady, which is Colette Shortt.
So I'm just going to take on where Jean-Michel left off. And I think, first of all, I just want to highlight that there were two aspects to the work that our task force did. One was the systematic review of the literature, and the second was a review of methodologies in this area, both current methodologies and new and novel methodologies. So we had two partner manuscripts ongoing. Another aspect that we really wanted to achieve with this work was to engage as many people as possible and to work cross-disciplinary, because oftentimes we're working in silos. So one of the aims of our workshop was actually to reach out to quite a lot of experts in microbiology, bacteriology, analytics, etc., as well as nutrition. And what was amazing is this workshop was held on the 3rd or 4th of December, just after the atrocity in Paris and during an emergency situation in Brussels. So I know Diana and the whole team were on tenterhooks as to whether we should actually go ahead with the meeting or not. And we decided to go ahead, and actually you can see from the graph here, we actually had a huge response to this meeting, highlighting, I think, the interest in the area across the disciplines. And I think secondly for me was how open people were in providing their expertise and their constructive criticism. We had, I think, 4 separate breakout groups, and we had some tremendous discussions. I think some of the best discussions that I've been party to since past claim, I would say, in Europe, in ILSI Europe. So here you can see we had NGOs, we had government, industry, and academia. Again, a key and salient point for ILSI Europe is to reach out and have a tripartite approach to all our work. And I think we were very successful in this regard here. So both manuscripts were critically reviewed. We had a tremendous networking opportunity and very constructive discussions. And we also had some presentations by some leading experts in Europe in the area. I guess we then came away with our drafts with lots of red marks. And we're at that stage now where we're trying to incorporate all of the comments. But what I'd like to do is give you some of the preliminary results that we can highlight from both the workshop and from our draft. We found actually quite few studies are focusing on the impact of gut microbiota on the metabolism of nutrients and non-nutrients, particularly in human studies. Quite a lot in animals, but not in humans, which I think quite surprised us. For proteins and digestible carbohydrates, there was no human study matching the selection criteria that we had. And given, I think, what Jeffrey mentioned about humanized oligosaccharides, that was quite interesting. Although we did see fiber-resistant starch and polyphenols were the most studied. And many commented on the 3 Ps, probiotics, prebiotics, and polyphenols as being sort of the top runners in this area. We found a lack of harmonization in the methodologies used, and this was over quite a short period of time. And this made it quite difficult to compare studies. And as Michel mentioned, in many instances, we had only one-off studies. And another point that came through was that there was a lack of consistency and specificity in relation to taxonomy and nomenclature. Just quickly moving on, with regard to proteins, we found that the main metabolites were ammonia, amines, phenols, and some branched chain fatty acids, and that was mainly from animal studies. In carbohydrates, we found butyrate, propionate, acetate, the gases, and biomass. And some of the studies, obviously, with butyrate went into the use of this as an energy source in colonocyte, etc. And polyphenols, which are not nutrients, but really depend considerably on the microbiota to actually ensure that bioactive substances are made, was probably was the largest area of where this research came through. Now, what we saw is obviously a diverse range of metabolites depending on the polyphenol that was actually fed into humans. Predominantly, we had studies on cocoflavanols, and we had metabolites such as equal and ferulic acid, etc. And a lot of the work highlighted the importance of these in human nutrition. About 5 to 7 phyla tended to be mentioned, and we have highlighted these previously. But one of the aspects that I just wanted to highlight was this inconsistency in taxa. So here you can see we tried to map the effect on energy on the different phyla. And here you can see that the Fecalibacterium in one of the studies was shown under the Fucobacteria, whereas actually that was reclassified around 2002 and now has been moved to the Firmicutes and should have been in cluster 4. So we found that it was quite difficult to actually interpret some of the papers in that respect. So just to finalize, basically it's a very challenging field of nutrition and health. And I think we need to include microbial logic and its kinetics in with our nutrition studies to ensure that we're getting the right answer to some of our questions. Obviously, as Michel had highlighted, it's a chicken and egg. Is it the microflora affecting the diet? But the diet also is affecting the microflora. And this is often, I think, forgotten, that we have a very, very dynamic situation ongoing. We feel that there was quite a lack of markers, and particularly that many of the markers, for example, with the short-chain fatty acids, these were being measured in the feces, but actually we didn't really have a handle of what was happening inside the black box of the body. And this is where our partner paper, which is going to look at methodologies, we hope will shed some light about new and novel techniques of how we get a more refined view. The other aspect is the harmonization of classification of microbes, or perhaps more importantly, the use of taxonomy and nomenclature and consistency in how it's used. And finally, also the importance of gut microflora and nutrition in drug metabolism. So from the workshop, one of the key aspects that came through quite loudly was the need for guidance in relation to microbiome studies in the field of nutrition. And that was, came across so strongly from the academics and experts and also from the workshop group that ILSI Europe has taken upon itself to put this forward as a new activity proposal. So just to finalize and to tie up with Jean-Michel's, let's discover a new country. And as we've already heard from the previous speakers, it's a very dynamic area, but I think we need to get some consistency and coherency into the methodologies that we're all using so that we're not duplicating studies that have been done previously and that we can interpret the work. Thank you.